SPARC webinar. We're pleased to have Professor Steve Beaudoin and Alan Smith discuss their work. This will be the last of our seminars, and we look forward to everybody participating in ADSA in November. And with that, both Steve's and Alan's bios are on the link. Please look to the link for detailed information. And with that, I'd like to hand it over to Steve and Alan.
This is a study that was assembled, we first saw it maybe 3 or 4 years ago. It was the brainchild of some of the folks who are actually in the audience for this seminar. Richard Leroux, Stefan Luco, Dave Atkinson were among the biggest supporters of the idea. And the idea was can we find a way to sort of develop not quite a standard, but develop a consistent reproducible method that the community could use to compare different kinds of traps for removing residues from surfaces that would separate the performance of the trap from the performance of the IMS for which the trap was designed. So we wanted to be able to do an apples to apples comparison between traps. And the point of most of what I'm going to tell you today was just developing the method and then to start to develop the database. And I'll preview for you a little bit of the ongoing study, which is going from the model systems that we used to go to some current commercial off-the-shelf traps and residues. I will mention that based upon the wisdom of the group I just mentioned, we then worked with the entire community to develop the set of standards that we used for this study, as well as the trap materials that we used for this study. Let's go ahead now and let me tell you about what we did here. Okay. So like I said, the goal was to investigate collection of residues from surfaces by a swipe testing. We wanted to separate the IMS from the trap. We selected 3 universities, Purdue, the University of Washington, and New Mexico Tech, to be the performers of a round-robin study. Signature Science was going to do the quantitative analysis to tell us about the results of the study. Transportation Security Lab was providing materials that would be used. The residues themselves, they were providing those for the study. And NIST provided us with the sleds that were used to apply the swiping loads during the sampling. I'll show you those in just a minute. And what we wanted to do was look for variability in all of the different aspects of the process. I'm not going to show you all of those, but we were looking for how reproducible is the overall process of contaminating a surface, recovering materials from a surface, and is it possible to really have a sort of standard assay. We wanted the protocol to be simple so the community could adopt it. And as I mentioned, we consulted the entire community to develop the materials that we used. Okay, so the slip peel tester that you see right here is less than $10,000. And this was going to be the unit that was used for the contact sampling studies. And what's nice about this is this platform right here moves across the apparatus at a constant speed. We can control the path of the swipe that's going over the surface of this sled. And we can also, over the surface of this platform, and we can control the angle of attack. And so we liked this particular apparatus. We had a custom designed sled that was used. This was going to be held in place by a cable that you can see down here in the bottom. And then the platform moves underneath the sled. Okay, and we were going to put a swiping material across the bottom of this, and you can see this, what I call the swiping nub right here. And the trap that's going to do the sampling is sort of affixed to that swiping nub, and I'll show you some more details about that. And the metal here is to provide a consistent load during the swiping, and you can see it's a 400-gram weight over the surface of this, and so the total downforce during this swiping was 6 newtons. Okay, so we had to have a very reproducible system, so I'm going to show you some of the guiding principles that we had, or the guiding equipment. So this is ABS plastic that we were going to put a residue on. And so we have a template, and you put the surface you're going to contaminate on the template, following the place substrate here instruction. Okay, and then you put this on a balance, and we are going to apply the residue using a swiping motion, a zigzag motion, and we're going to follow a path. We have a template for the dry transfer. We're going to follow a path like this and apply a load that we specified so that we can get a reproducible surface, a reproducible residue on the surface from lab to lab to lab. And New Mexico Tech, I should point out, designed these templates. And as I mentioned, we had a 10-centimeter contamination path or sample transfer path, and we were using a zigzag motion with a well-defined downforce. We went always at 300 grams. Then we had to now move the Now we put you can see right here that we've got the template. We're going to have our contaminated surface on the template. This platform is going to move, and we're going to have the sled sort of sliding across the platform as it moves. We have a protractor so we can make sure that everything is lined up nice and perpendicular to the direction of motion. We have the height of the cord that is attaching the sled, holding it in place, so we have the angle of attack maintained. And we designed these aluminum bumpers to keep the sled from translating. We only want it to translate in one direction. And so now you can see the completely set up apparatus. So as I mentioned, we custom fabricated these bumpers, a bowling analogy, to keep the sled right where we wanted it. And then we had our surface that was contaminated with residue held in place, and then the swiping sled moves across that surface, as I just mentioned. Okay. We used ABS plastic, uncoated aluminum, and nylon as the three substrates that we were going to contaminate and study removal. These were all well-defined sizes. They were all prepared so that if there was any pattern in the substrate, that pattern was always oriented in the same direction relative to the swiping. In terms of the sled, you can see that it's in multiple pieces. And so you can undo this set screw here, take the sled apart, and you can install, and we installed aluminum foil that covered this swiping nub and then was held in place up inside the body of the sled here. And then it went down and covered that swiping nub. And then over the surface of that foil was the trap that we were using to collect residue. The point of the aluminum is that anything that made it through the trap would be collected on the aluminum, and both the aluminum and the trap were assayed for total recovery of residue. And as I mentioned, the load was defined by the weight of the sled, and we had a very precise path length and a precise swiping speed. The residues that we collected, which are on the trap and on the aluminum, were placed in these centrifuge tubes, which were sent back to Signature Science. They went into the tubes. We put ethyl acetate, a consistent amount in every tube, and sent that to Signature Science for assays. We also sent the dry transfer strips back to Signature Science. We knew exactly how much residue arrived on the dry transfer strip from TSL from their very well-defined protocol. And then by sending the spent transfer strips to Signature Science, we could look at how much material was transferred onto the surface we were contaminating. It was a lot of fun to develop this method of sending these residues inside this solvent. We found every imaginable way to not properly seal the top here so that solvent was all over the place and so that the labels on the samples were dissolved. But eventually we figured out what we were doing, and we were able to get the samples off to Signature Science in a reproducible method that didn't fall apart in transport. Okay, so we had three swipes that we used. One was this tech swipe material. The tech swipe is a common clean room cloth that's used for cleaning surfaces in clean rooms. It has a side-dependent pattern. One side is herringbone over here, and one side is ribbed. We always set these up so the herringbone side was used, and so the same orientation of the herringbone towards the surface that we were swiping was maintained. We used a stainless steel mesh as the second surface, as the second trap type, and the stainless steel mesh also has a pattern to it, and we oriented the swiping of the surface so that we were always moving perpendicular to the pattern. The last trap that we used was muslin, which was isotropic in terms of the pattern. And so we didn't have any particular orientation to the surface for that. We used two threats, RDX and C4. As I mentioned earlier, we had an aluminum substrate, ABS plastic, and nylon. All of those were purchased from the same lot, so they had the same surface finishes, the same weave, as much of the characteristics as possible were the same. There were 10 replicates for each residue on each surface with each trap conducted at each of the three performers. So there were 180 experiments conducted on each threat substrate surface. And those were all evaluated at Signature Science. And I'm going to turn over to Alan in just a moment. He's going to talk about the wet chemical analytical method that they developed. And so, Alan, can I turn this over to you right now? And I'll advance the slides for you.
Sure thing. So once we received the samples, we took the 15 mil conical tubes and they were vortexed for just a little bit to make sure everything is settled properly after shipping. And then we put them on a rotomixer for 15 minutes just to make sure that everything was treated the same and extracted as efficiently as possible. We then took the ethyl acetate from the 15 mil conical tubes, and those are transferred into little glass test tubes. They're 12 by 75 millimeter glass test tubes. They're about 3 inches long. And then each sample extract was evaporated down using an antivap, so just a stream of nitrogen, all the way down to dryness. We kept an eye on them to make sure that they weren't kept dry for very long to avoid any sort of volatilization of the explosives that were at the bottom of the tube. And then the samples were reconstituted into 1 mil of acetonitrile. The reason for that solvent exchange is that at the beginning of the TESSA-1 project, we did studies looking at the chromatographic performance of the various extraction solvents that were available to us, and acetonitrile performed better than ethyl acetate from a chromatographic standpoint, but ethyl acetate would extract better. So we used ethyl acetate as the extraction solvent, but then did a solvent exchange over to acetonitrile prior to analysis. And after reconstitution, we put the samples into just a standard 2 mil amber vial and analyzed them by LC-MS. Next slide. So we analyzed the samples on our Agilent 1290-6410 ultra high pressure LC-MS, so it's a triple quad instrument. Our LC solvents were relatively standard for explosives analysis. Our aqueous phase was 0.25 millimolar ammonium nitrate in water. Our organic phase was 0.25 millimolar ammonium nitrate in acetonitrile. We used a Phenomenex Gemini C18 column and all the other various parameters of it are shown there, with the column being held at 40 degrees C. Samples were ionized by APCI. Our solvent gradient is there on the right side of the slide. It's relatively standard. We started at 80% A and go to 5% A, and then kick back to 95% A just to make sure we wash everything off we possibly can and then re-equilibrate at the end. The MRM parameters are shown below. There's more than the targets that we used on this particular study, but when we originally designed the MS method, we wanted to make sure that we could see all the nitro explosives that might be used in this program. So this method does work for TNT, PETN, and RDX. Also included is an isotopically labeled RDX. That was an internal standard that was used in every analysis. It allowed us to normalize for any instrument or extraction specific variability that may have happened during extraction. So that's why that was included as well. So shown here is just some of the instrument performance. Above is what the MRM transition looks like for RDX at 5 nanograms per mil. So that is the lowest point on our or the second lowest point on our calibration curve. We have what I call relatively reasonable signal to noise ratio, and no other peaks are shown in the chromatogram, which is to be expected when you're doing MRM analysis. And then below is a calibration curve example of RDX running from 1 nanogram per mil up to 500 nanograms per mil. The spike amount for this study was 250 nanograms per mil, so we never expected to see anything above that, but the curve was relatively linear across the entire range with an R squared of 3 nines. And so almost every curve that we ran over the months and months and years of this analysis looked exactly like this. And so when we analyzed the samples, we tried really hard to normalize and standardize the order in which they were analyzed and what each sequence looked like. So this is kind of an example of how the samples were laid out in the work list or analytical sequence before they were put onto the instrument. So at the beginning was our calibration standards running from lowest to highest. The curve shape had to be linear or quadratic. I think the majority of the time it was linear for RDX. The R squared had to be at least 2 nines to be accepted, and I think that was never an issue. And the accuracy over all the points had to be plus or minus 30%, so between 70 and 130. After we ran the cal curve, we injected all the QC samples that were included. That would be negative control samples, blanks that were prepared either by the laboratory that sent us the samples or prepared at Signature Science, and any positive control samples that were prepared either at the Performer Lab or at Signature Science. We included various method blanks and trip blanks to ensure that no contamination had occurred either at the collection site or during shipping, and then we included method blanks on Signature Science's end and extraction blanks to ensure that no contamination had occurred at our site or during sample extraction and running them on the instrument. And the positive control samples should help verify that extraction efficiency was still in the high 90% as we had seen during method development and had seen during the entire program running. And then we would analyze 10 program samples. So each batch of 10 swipes that a performer sent to us was actually 30 samples. There were the 10 Teflon strips that the TSL had created with the explosive put on them. Then there were the 10 swipes that were collected off the surface, and then the 10 aluminum backers that were sent to us as well. And so based on how much RDX was recovered from each of those surfaces, we could accomplish a mass balance of knowing where the RDX was and wasn't, and we could determine collection efficiency, transfer efficiency, and stuff like that. So the 10 program samples, what we'd usually analyze first were the Teflon transfer strips from the TSL. Then we would run a QC insertion with an SVS or a sensitivity verification standard. That would tell us is the instrument as sensitive as we expect it to be? And then we run a CCV, which is a continuing calibration verification standard, which would tell us is the instrument running at the calibration that we expect it to. As long as both of those passed, we could keep running. And then the next 10 we would run would be the actual swipe extracts. And then at the end, we would run the aluminum backers. As we went through the project, we started to figure out that no analytes were breaking through the swipes themselves onto the aluminum backers. And so we started analyzing fewer of them just to give us more analytical throughput. Steve, you can go to the next one. Thanks. And so then after the samples were analyzed, we had this automated reporting system that we had generated inside using R scripting. And so we would manually generate some of the metadata for each sample set and then take some of the automatic outputs from the Agilent software itself. And we would create this multi-page report that could go to both the performers, actually to all the performers and to Northeastern as well, to show this is what we saw in this batch. So the metadata is there in the top right. That's a very small portion of the metadata that would be associated with each sample set. And then on the bottom is kind of an automatic output of what we got from the R script showing, who sent us a sample, that's the performer, the substrate that they used, the swipe, which transfer strip, how much was left on the transfer strip, how much was on the stainless steel mesh, how much was on the aluminum foil, and then essentially a total recovery based on how much we expected to get, and then a dry transfer efficiency based on how much was actually left on the Teflon strip and how much and what the difference between that and the spike amount was. And so we got all this data for each sample set, and those were all compiled at the end. And I think there's one more slide from me, Steve. Yeah, so then each report contained kind of a summary of results in text form. We would type that up manually. A summary of results table showing peak areas and concentration. That's the top right there. the mass balance for each swipe and Teflon strip, that was on the previous slide. The QA data for each sample set, so were there hits in the blanks, what was the extraction efficiency if there were any spikes included, and then a complete output of all the sample metadata included in each sample set, and that's that bottom right insert there.
All right, thanks, Alan.
You're
So
welcome.
I'm gonna jump up for just a second to just draw your attention over here to some typical results. You can see dry transfer efficiency, getting the residue off the dry transfer strip onto the onto the surface to be contaminated, was generally pretty high. And swipe collection efficiency was generally not. And you can also see that there was a lot of variation. You know, here's ABS plastic, and we were putting RDX on ABS plastic, and dry transfer efficiency ranged from 52% up to 90% sample to sample variation, and the swipe collection efficiency varied from basically nothing to about 12%. So I call those to your attention as we move into some results, and I could show you graph after graph after graph. Alan provided me no shortage of data, but all of them look exactly like what I'm showing you right now. Okay? So here you see the results from New Mexico Tech, RDX removal from ABS plastic, and you can see with TechSwipe or the muslin or the stainless steel trap, there were changes in the collection efficiency, but it was not consistent, the trend for New Mexico Tech, not consistent with the trend from Purdue. New Mexico Tech had a great day recovering material with TechSwipe, not so good with the muslin. Purdue, on the other hand, didn't do very well with TechSwipe and had a great day with the muslin. Washington was pretty consistent between the two. Everybody, no matter what residue we were looking at, no matter which surface we were looking at, had a bad day with stainless steel, no matter what. Muslin and TechSwipe are generally comparable, but the relative effectiveness varied sample to sample, lab to lab. If we average everything out, we got somewhat consistent results, but very, very large error bars. So this is ABS. This is the RDX again, and now this is off the aluminum surface, which was a little different roughness. And interestingly here, the stainless steel actually performed really well for New Mexico Tech off of this aluminum, which is very unusual. But the aluminum was very difficult in certain cases to recover from. The nylon, on the other hand, while with TechSwipe we did a pretty good job on the nylon, in general, if you look, the overall recovery from the nylon was always poor. And there was quite a bit of variation. So we looked at the characteristics of the trap, the characteristics of the surface, the lab-to-lab variations. We could not find any consistent correlation with any of these features. It was always very large variation in recovery within a lab, very large variation from trap to trap, and very large variation from surface to surface, with one exception. And that one exception, so here's what I just said to you. What we could pull from it is that stainless steel trap was generally the worst performer. Stainless steel did not have surface topography that was capable of interrogating variations on the substrate. And so that didn't perform particularly well compared to any of the traps overall. Nylon in general was the toughest material from which to recover residue. This is because the nylon had the most open topography, and there were the most locations on the surface where the residue could effectively hide from the trap that came across the surface. So in general, nylon was always the toughest material from which to sample. But we did see one interesting sort of result, and I'm showing that to you here. This is the collection efficiency as a function of the standard deviation in the RMS roughness of the surface from which we were removing material. And what we finally see here is there is some correlation. So you can look PETN for the surface that had the smallest standard deviation in the roughness. We had the overall best collection efficiency, and this is averaged over all traps in all universities. PETN, the smoother surface or less variation in the roughness, gave us the best recovery. More variation in the roughness, poorer recovery. Most variation in the roughness, worst recovery. The same held true for the RDX and for the C4 and for the average of everything in gray. Okay. The same was true if we went ahead and looked at the peak to peak height. And so this is the tallest peak, the difference between the tallest and smallest peaks on the surface, which again, it's the standard deviation in that height. If we plot the collection efficiency as a function of that standard deviation, we see the same trend, a dramatic reduction in the collection efficiency as the deviation in that peak to peak height changes. So what this suggests to us is that the randomness of the topography on the surface is what's controlling in the recovery. Okay, I got you, Kristen, thank you. And so the way that I describe this is, imagine, I hope that all of you have been in a place where it has snowed. So imagine that you're snow plowing the parking lot. The standard deviation in the peak to peak height or the standard deviation in the RMS roughness really is all about the roughness of that parking lot and the randomness of the size of the holes in that parking lot. If you're trying to snowplow gravel versus trying to snowplow something that's, you know, fresh asphalt, you're going to recover a lot more material from the fresh asphalt than from the gravel, because the gravel provides many locations on the surface where, as you're trying to push the residue across the surface, whether it's snow or explosive residue, you're pushing the material across the surface. And if there's lots of non-uniform features on that surface, the residue can just simply hide there, and you can no longer extract it with your trap. And so that is what we think is what was controlling the behavior that we saw here and leading to these very irreproducible results. It all came down to the irreproducibility of the surface from which we were doing our sampling. And so with that said, I'll just conclude quickly. You know, we defined what we thought was a very tightly defined sampling protocol. We thought very good control over how we deposited the residues and harvested them. The extraction and quantification, I thought, was exquisitely precise, but yet we saw these enormous variations in both the deposition and the removal, and this seemed to correlate with only the variation in the topography on the surface that was contaminated. We're continuing our study now, and we're moving forward to look at a slightly expanded set of residues. We're brought in Semtex A, and now we're using some commercial off-the-shelf traps that are currently qualified for use in air transportation security environments. We have the tech swipe as the carryover, so we have some consistency to the prior study, and we're using Lincoln Labs as some chemically active traps, and we're going to involve them as well to see how those work. We are just at the beginning of this particular study. COVID got in the way and shut us down for about 8 months. But I'm looking forward to sharing these results to you as they come online sometime next year. So with that, I acknowledge the support of ALERT, the Department of Homeland Security, Science and Technology Directorate. Thank you for your attention, and I'd be delighted to take any questions if we have some time.